another 2 minutes, sir. So with all your permission, we'll start by 2 minutes. Krishnangshu sir, can you open your video, sir? Krishnangshu Rai sir, আপনি কি ভিডিওটা অন করবেন স্যার? ভিডিও অডিওটা একবার দেখে নাও। Yes sir, yes sir. Okay.
I think it's audible now.
Yes sir, audible sir.
Okay. Right.
Good evening, sir.
Good evening. How are you?
Fine, sir.
Huh? You have doubled your volume of your body.
Yes, sir. Yes, sir. Yes, sir.
This is a sign of prosperity, it seems.
I don't know. I don't know.
And about your new mustache. Congratulations.
Thank you, sir. Thank you. I've tried to reduce the size of my mustache. Yeah.
What happened to your teeth?
Yeah, it is like that only.
Like that only. Okay. Great. Any change on me, you find?
No. You're looking trimmer.
Trimmer. Okay. Thank God.
Trimmer and slimmer. Yes.
God bless you.
Yeah.
Yeah.
Okay. Good evening, everyone. So, on behalf of Indian Pharmacological Society West Bengal Chapter and Inovocare E-Academy, I welcome you all in this academic fest. So, today we are really fortunate to have two stalwarts in the field of pharmacology and clinical pharmacology with us as chairpersons, Professor Krishnangshu and Professor Santanu Munshi. And also we are really fortunate to have one of the noted pharmacologists in the country, and we all are really privileged to have Dr. Viraj Suvarna with us, who needs no much more introduction. So with that, I am requesting our respected chairperson sirs to please introduce today's topic, that is development of biosimilars in Indian perspective, and then we can start today's proceeding. So may I request our respected chairpersons to carry forward today's meeting.
Okay. Good evening, Shambo.
Good evening, sir. Good evening.
For your kind words. And it's interesting to see Dr. Munshi was my colleague once. We worked together and we had a nice time in School of Tropical Medicine. Of course, before that we are introduced and had a friendship. And nice getting introduced to Dr. Suvarna. So Dr. Suvarna appears that you are quite younger to us. So nice brother from the same fraternity. And I have heard about you, about your escalated CV and your activities, and nice that in the evening we are getting you to have our knowledge refreshed. The subject given to us today by Dr. Shambo on behalf of the aegis of Indian Pharmacological Society was a very challenging one, you see. The term, the biologicals and the biosimilars, so these terminologies came, so I think it's about 10 years back from now. It was a virgin terminology. It was when I was doing clinical pharmacology, professorship, administration in my service, earlier service, it came. Then what was the meaning of it? How we'll define to this? That was a debate those days. How does it differ from the generics? That means the drug which has been approved by FDA. How does it differ? And what are the benefits? Why they are in the market? What is the necessity of those subjects and what for they are indicated? These are the subjects to be discussed. And I believe Dr. Suvarna is the best person because he had a lot of experiences I see from its CV that the drug development field, a drug is developed from lab to market. It takes long time, huge amount of money, scientificity. So all these things, he is the right person, and I thank and congratulate Dr. Shambo and his group for organizing such a nice seminar in this evening, so that all of us will get enriched by your knowledge. I just told about this academic fest. It's really an academic fest. And I'm also happy that Dr. Munshi, Professor Munshi will be giving his inputs, because he is days in and days out of working with the clinical pharmacology, drug development and all these. Of course, I'm not now directly related to pharmacology these days. I'm doing administration full time. Anyway, I like to hear from Dr. Suvarna, Dr. Munshi, their inputs. So before I request Dr. Suvarna to speak, I request Dr. Munshi to give his few words, introductory remarks, then we'll go to the subject proper. Thank you very much.
Thank you, sir. Professor Krishnangshu is one of the stalwart in pharmacology. And thank you for your kind words. And I happen to know Dr. Suvarna for a pretty long time. I've been following him since his days in Pfizer. And he has worked in different therapeutic areas. And now he has taken up a new challenge in Biocon as Vice President. And he is also the medical affairs head. And biosimilar is the next big thing for India. And in 2020, a lot of molecules are going off patent, have gone off patent. So the opportunity for biosimilar is huge. And India is becoming the global capital for biosimilars. And without wasting any time, I would like to hear Viraj on this topic, biosimilar development in Indian context. Welcome, Dr. Viraj. Please take over.
Thank you so much, Dr. Munshi and Dr. Ray, and of course, Dr. Shambo and the entire Indian Pharmacological Society West Bengal Chapter. It's an honor for me to be among stalwarts. And while Dr. Ray mentioned that he would be enriched, I feel I will be enriched with the interaction that we will have as soon as I finish my presentation. So let me now just share my screen. And can you see Shambo?
Yes, sir. Yeah.
I'm just putting it on slideshow. Yeah. So today's topic, very interestingly, when you see my presentation, I won't be talking too much about, when you look at the slides, they're not really slides based on biosimilar development in the Indian context, but they'll give you the context against which then I will talk about biosimilar development in India. So I have mentioned here, of course, that the Indian guidelines came out in 2012. And that's pretty good because the first international guideline on biosimilars came out from the European Medicines Agency. This was in 2005. So they are the strictest. And then, of course, we revised our guidelines in 2016. And I'm sure going forward, there could be another revision as well. So I will basically focus on, what Dr. Ray was saying, that, what really is a biological product in the first place. Then, of course, talk about the biosimilar, then differentiate it from, say, a generic, and talk about the development pathway for biosimilars. And as I'm doing that, I will kind of compare what happens, say, in the European Medicines Agency or the US FDA, vis-a-vis what happens in the DCGI's office. And I'll also give you my own perspective on the reasons why our guidelines perhaps are not as stringent as those in the European Union or the US. So first, of course, what is a biological product? And that I'm sure we know that generally they are large, complex molecules which range in size and complexity, but importantly, they are produced in living systems, so microorganisms, plant or animal cells. And the technology is important, recombinant DNA technology. And in this presentation, obviously, I'll be referring to not just the original reference biologic, but also biosimilars. And another interesting term, which happened actually only last year on the 28th of July, interchangeable biosimilar. And interestingly, an Indian company was the first to get this designation or approval from the US FDA for its biosimilar insulin glargine. And this is a matter of pride for Indians, because imagine so many MNCs are also into biosimilars, and yet an Indian company, I don't have to name the company because I'm sure you all will know which company I'm talking about. An Indian company managed to get interchangeability for its biosimilar insulin glargine for the first time in the world on the 28th of July, 2021. And of course, it's an exclusive kind of thing for 1 year, after which other biosimilar insulin glargines can also get this interchangeability. But first, let's try and understand what really is interchangeability, and I'll come to that a little later. Here, of course, what you're seeing are some examples of biological products. So, what our body produces: insulin, erythropoietin, and also the monoclonal antibody. Now, when you come to a biosimilar, so all of us know about small chemicals, and when the patent on the innovator or originator small chemical expires, then generics come in, which are again small chemicals only. They have to only show that they are chemically equivalent, which is not too difficult, and bioequivalent, that means pharmacokinetic equivalence. But they do not need to show clinical or therapeutic equivalence, because it is presumed for small chemical generics that if they are chemically and bioequivalent to the original small chemical, then clinically or therapeutically, they will be, I can't say the same or equal, but similar or equivalent. And naturally, once the patent for that original small chemical expires, we need more people to have access to that medicine. And therefore, even the regulators will not expect the generics manufacturer to do too much in terms of development, because then naturally the cost of that generic will also go up. So that is understandable. Now, similarly, when a biology goes off patent, and as Dr. Munshi was saying, quite a few biologics have already gone off patent, and many more will go off patent very soon. So naturally, many Indian companies are, already preparing for that, what they call it, the patent cliff. So lots of biologics, when they lose their patent, the Indian companies and of course other companies in the world also would want to be the first biosimilar to get approval. So, and it takes time. So they have already started their preparation. So essentially what a generic is to a small chemical innovator, a biosimilar is to an original reference biologic. But naturally, the biosimilar is not that easy to make. So it's not like a small chemical. And then if you look at the development pathway, you'll realize that there is much more that the biosimilar manufacturer has to do. And that is why when you look at the price differential between the original reference biologic and a biosimilar, it is not that much as compared to perhaps even an 80% price differential between a small chemical innovator and its generic. But clearly the biosimilar should be proven to have no clinically meaningful, which means obviously you have to do a clinical study, no clinically meaningful differences in terms of safety, purity, and potency. And then they go on to talk of safety and effectiveness. Of course, immunogenicity is also extremely important. Biosimilars are made from the same types of sources, are administered the same way, have the same strength, dosage, potential treatment effects, and potential side effects as the original reference biologic, and all US FDA approved biosimilars undergo a rigorous evaluation to ensure they are as safe and effective as their reference products. But now when you look at these three, not squares, maybe rectangles, you find one very clear difference. When you look at the reference product prescribed by a provider, biosimilar prescribed by a provider. But when you come to interchangeable, there it's written prescribed by a provider or maybe substituted at the pharmacy without the intervention of the prescribing provider. Now, this is a little strange because it doesn't happen elsewhere in the world. But in the US, if the FDA gives a product interchangeability, what it means is, for example, suppose I take insulin glargine, which is Lantus, which is the original reference biologic from Sanofi. And suppose I talk about Biocon Biologics and its own insulin glargine biosimilar. In India, we market it under the brand name Basilog. In the US, we use a partner called Viatris, and they co-market the same product, but under a different brand name called Semglee. In the US, suppose a prescription of Lantus comes to a pharmacist, and suppose Semglee has got US FDA interchangeability designation, the pharmacist can substitute and he does not even need to inform the doctor. And this I find a little strange because in India, of course, it will never happen, and it should not also because the doctor should know. But anyway, interchangeability is therefore, as you must have realized, it's a one step ahead of just being a biosimilar. So when you're a biosimilar, what it means is the doctor has a choice. Either he writes the original reference biologic, for example, Lantus, or he can write Basilog. But if interchangeability is also established, then a patient who is currently on Lantus can actually be switched by the doctor from Lantus to Basilog. This is because a specific study, a specific design has been used, and only based on that, this interchangeability approval is given. So we'll come to that. But before that, this, of course, we are aware of that there are differences between small chemicals and biologics in terms of how are they produced. So one is chemical, one is a living organism. Then in terms of characterization, so obviously for a biologic, it's more comprehensive. And because of the sheer size, it's difficult to give it orally, so it has to be given parenterally. And then in terms of manufacturing, again, it's more difficult. And lastly, when you look at safety, immunogenicity is important. And this I'll tell you why. Now, we all know that to a biologic, our immune system can produce antibodies. Now, as long as the antibody that we produce, the titer, is the same as what we produce to the original reference biologic, it is fine. But suppose a particular biosimilar, say in this case insulin glargine, there is more antibody development by the patient against this biosimilar. How can it affect the patient? So one is, of course, it affects in terms of efficacy, safety. So then the doctor will stop the biosimilar insulin glargine and naturally shift the patient to the original reference biologic, in this case, Lantus. But those anti-insulin antibodies can also destroy Lantus. So therefore, even Lantus may be ineffective. And that's the reason when you select a biosimilar, please only select a biosimilar where you know, one is the company, of course, behind the biosimilar. So does it have extensive experience and expertise in not just manufacturing the biosimilar, but also getting it approved by international strict regulators based on good quality clinical data? Then obviously you're on a good wicket, but otherwise you're taking a risk. Now here, all that I want to mention about are certain terms. One is, of course, biomimic or biocopy. Now this is the term given by the US FDA to a particular, suppose, as a manufacturer, you submit your dossier and the US FDA finds that you are not similar. So then they give you a particular designation, mimic or copy or non-regulated or intended copy. And if you go to the European Medicines Agency and a similar thing happens, they will straight away call you a non-similar biotherapeutic product. So they are very clear. Now, when I look at bio-better, naturally, we may think, and we are right when we think that when I do that clinical study, comparing the biosimilar to the original reference biologic, it may happen, although we are doing an equivalence or a non-inferiority design, it may happen that the biosimilar may turn out to be better. Now, in such cases, yes, of course, the term would be bio-better, but why nobody wants to call their molecule a bio-better is because from a regulatory perspective, a bio-better is looked upon as a new biological entity. And then that company would have to repeat the entire clinical, or not just clinical, preclinical and clinical development program that the original reference biologic had to undergo. And naturally, no company would want to do that because, it takes a lot of time, effort, and money. And especially at this stage of the life cycle of the product, no one would want to do that. So there are very few examples of such bio-betters. But one thing I can tell you is the company would not want to even call it a bio-better. Although, that study has clearly shown that was better. I think there are some examples with trastuzumab. There's trastuzumab deruxtecan and trastuzumab adoemtansine, which were found to be better, but they're not marketed as bio-betters for this reason. Now, this is just to give you an example that why I said the European Medicines Agency is very strict, because they actually have rejected certain applications and asked the company to withdraw. So one was a company called Marvel, which was coming out with insulins. Then, of course, interferon alpha, so Alphion. So there are some examples where clearly the EMA, that's why I keep saying EMA is the strictest. But the best part is it's not only with the biosimilar, even with the biologic, they're very strict. So let's understand that J&J had come out with an erythropoietin alpha called Ebrex. And in good faith and intent, they replaced human serum albumin with sorbitol 80 stabilizer using uncoated stoppers in their pre-filled syringe. Now, they did not know, but this led to an increased incidence of neutralizing antibodies and a particular adverse event called pure red cell aplasia. In fact, it was even published in the New England Journal of Medicine. And what I'm trying to get at by giving this example is to tell you that with biologics, there is inherent variability anyway. So you need to be consistent, batch to batch. And that is why, again, I'm coming back to the same thing, that this company whose biosimilar you select should be a company that has been in the business of making biosimilars for years, biosimilars and biologics for years, because then it would have that, consistency, batch to batch consistency. But that is very important for biosimilars as well as biologics. And then, of course, why immunogenicity is important. So I gave you one example, but there are other examples. So a somatropin biosimilar, then, of course, an erythropoietin, and that I've already spoken about, thrombopoietin. This is interesting, recombinant human thrombopoietin, which can actually produce platelets in your body. So I know we have thrombopoietin receptor agonists like L-thrombopag, and then, of course, there's another product, romoplastin. But years ago, I think it was Genentech, or was it Amgen? They came out with pegylated recombinant human megakaryocyte growth and development factor, but it was highly immunogenic, and it actually led to paradoxical thrombocytopenia. And obviously, the development program was stopped. Sorry, the other product was romiplastin, which is nothing but I would say truncated version of pegylated recombinant human megakaryocyte growth and development factor. Then there's something called as a tryptophan eosinophilia myalgia syndrome. So, the point I'm trying to make is with biologics as well as biosimilars, these things can happen. And therefore, you need to go with a good company rather than a company which only gets their biosimilars approved in India, and they are not really in favor of going abroad, because naturally when you go abroad, it's not that easy. In India, I'll come to that a little later, it's not very difficult to get a biosimilar approved. But if you read the guidelines, they do place a lot of emphasis on the very first step, which is chemistry, manufacturing, and controls. But thereafter, when we come to the clinical study, the phase 3, Unfortunately, there I think they're just saying 100 patients in each arm. And when you do equivalent studies or even non-inferiority, sometimes that 100 in each arm may not be adequate. So if your study is not adequately powered to pick out a difference, then you may conclude by saying there's no difference between the biosimilar and the biologic, and you'll get approved as a biosimilar. And all that you need to do thereafter is a PMS, or no, I think a phase 4 interventional study on 200 patients. That's it. But my thought was that what if there are differences, but you were not able to pick it up just because the sample size was inadequate or the power? So that is the only area that I feel the Indian guidelines can be updated or upgraded. But the reason why I think they're doing it is because they also want companies to come out with biosimilars at a price point where many patients can access. So that is good. But at the same time, what if a particular biosimilar is highly immunogenic and they have not been able to show that it is similar in immunogenicity to the original reference biologic? Then it can create problems, and then the cost can go up. So one has to look at overall cost also, rather than only the acquisition cost or the price that you're paying when you purchase that particular product. Now, this is again just to give you a little more on immunogenicity. Some are patient-related factors like the underlying disease, immune status, concomitant medications, genetics, age, dosing schedule, route of administration, and previous exposure to related proteins. And product-related could be the process of manufacturing, glycosylation, degradation products, the formulation itself, structural homology, post-translational modifications, I'll come to this a little later, chemical modifications, impurities from host cells, and then, of course, stability. Now, this is an example of recombinant DNA technology. Now, I remember when I was doing pharmacology, my professor, she told us what is the most simplest definition of a drug. And she said a drug is something that is given, or that when administered, it alters existing function of the cell. It does not give new function to that cell. But now I'm thinking maybe that definition needs to change, because recombinant DNA technology has made a bacterial cell or a yeast cell produce something which its grandfathers also never produced. And as you can see here, all that we have to do is identify the gene which codes for that protein of interest, take that gene onto a plasmid like a vector, introduce it into either a bacterium like E. coli, or there are yeasts like Pichia pastoris, or Saccharomyces cerevisiae, or Hansenula polymorpha. And then it integrates into the host, or rather that bacterial or yeast cell, in the nucleus, in the genome. And then obviously, as all of us know now, because of the pandemic, you have DNA, transcription, then RNA, and then how the entire thing gets translated. So you have messenger RNA, then transfer of soluble RNA, and then obviously ribosomal RNA on the rough endoplasmic reticulum. And that's like your protein factory, where these proteins of interest are produced. And when that cell divides, the daughter cells also get the same information. And that's how you have a whole cell bank, a master cell bank and a working cell bank. So that's the way recombinant DNA technology works. This is a similar kind of what I said earlier, but here I'm just trying to also tell you that sometimes a particular yeast, like what Biocon uses is Pichia pastoris, there are some advantages, as you can see in this rectangle. But essentially, the process is important to know, so that we understand that recombinant DNA technology is resorted to. And as you know, naturally, the origin, the starting point that the innovator uses, it is proprietary. They will not share it. So naturally, the biosimilar manufacturer has to use a different starting point. They arrive at more or less the same products. The basic amino acid sequence or whatever would be the same. So to that extent, you might want to say, are they bio-identical? But the reason why we don't say bio-identical is because in this process, as I mentioned earlier, post-translational modification happens, like glycosylation or sialination or acetylation, amidation, phosphorylation. And because of all that, I can't really call them identical. And therefore, the word used is similar And then you need to, of course, do a clinical study also to prove that there are no clinically significant or meaningful differences in efficacy, safety, or immunogenicity in adequately powered, well-designed studies, phase 3. So then you can call it a biosimilar. And this is just, again, to take us through the process of the gene sequence, putting it inside the vector inside a cell, fermentation, purification, and then you get your highly complex protein. And that is why, in fact, with biologics or with biosimilars, this word is always used. The process is the product. Now, with Biocon Biologics, one interesting aspect is that the drug substance is produced in India only. That means, see, we are not dependent on, say, China or any other country. Because if you're dependent on, say, for example, China, we know what can happen between India and China. So at any point in time, if something happens there and then you are dependent on them, you don't have your API or drug substance, you can be in trouble. So therefore, it's important for all companies to have their own drug substance manufacturing facility, because then you're truly, I would say, self-reliant or truly independent. And then, of course, your manufacturing facility will be inspected by US FDA, EMA, wherever the product goes to, whichever country or countries. This is that variation that I was talking about. And therefore, with biologics or biosimilars, we need to be very consistent in the way we manufacture. And that is why we have to go by experience and expertise. We know that biological products are larger, complex, and proteins, as you know, I think we learned this in school also, primary, secondary, tertiary, quaternary structures. Then, of course, they're glycosylated, there are more than one isoform. All of this can affect PK as well as PD in terms of dissolution. And then, of course, the ADME or ADME, and then even the interaction with the receptor. Then, of course, cleavage of terminal amino acids can activate a protein. Glycosyl groups play a key role in signaling and can influence blood clearance rate. Attachment of lipids allows protein to be anchored in plasma membrane. So all these different changes can happen. And therefore, it's important that you can't just stop with chemical or bioequivalence. You need to do further studies. And that's why it's not that easy to come out with a biosimilar. And how is it different? We've already seen this. The process is the product, the larger size, sequence, and structural complexity. Proteins are less stable. Safety and efficacy dependent on robustness and monitoring of quality aspects, and obviously the potential for immunogenicity. And why do we care for this? Is obviously we know that these antibodies, imagine sometimes you can get an antibody to an antibody. That means you give a monoclonal antibody, and to that antibody, your body can produce an antibody. I mean, it's very strange, but these are things that can happen with biologics. These are some of the consequences when I say safety, not just anaphylaxis, but neutralization of the endogenous protein or even death, altered efficacy in terms of exposure, neutralizing anti-drug antibodies, and even the clearance, sometimes even the absorption, the assay interference. And this, of course, we've seen earlier the differences between small molecule and biosimilars, so I won't belabor the point, except to say that it's obviously more difficult to make a biosimilar than a generic. And that is why to see a biosimilar that is as cheap as a generic, I would be a little suspect, because there is not too much of a difference in price between the original reference biologic and a biosimilar. But with a generic and small chemical, there can be, as I said, even an 80% price differential. And we know why biologics cannot be treated as standard generics, because there are differences. And these differences can lead to major changes in safety, efficacy, and also immunogenicity. Now, here, the terms I want you to focus on are not just the fact that it will be a biosimilar, but is it interchangeable? That I've already explained to you. Is it substitutable? And this is what happens at a pharmacy level. Pharmacovigilance, I have a separate slide on it, so I'll talk about it. And then, see, for a biosimilar, it's not that they have to do studies on all those indications. Like, for example, a Humira, adalimumab is indicated in many indications. But that doesn't mean the biosimilar of adalimumab will have to get approvals by doing studies in all indications. No, they will select maybe the most important indication, which is the most sensitive in terms of the endpoints and even the patients. And then ultimately extrapolation is allowed because, obviously if you make the biosimilar manufacturer do exactly what the original reference Biologics did, then the price differential will not happen. And the whole purpose of coming out with generics and biosimilars after the patent on the innovator expires is so that more patients, indicated patients, can access the product. Prescribability, that is where you just show biosimilarity, so it is prescribable. So here the international non-proprietary names are different, but switchability or interchangeability is where you have proven therapeutic equivalence by doing a specific kind of design, which is called the 3-switch design. Then of course it can be approved with the same INN, except that now it's not just biosimilar insulin glargine, for example. You have a 4-letter suffix, like in the case of Basilog, it's YFGN. So in the case of other biosimilar insulin glargines, they'll have their own 4-letter suffix because we have to differentiate. Each one has its own fingerprint. And when you're reporting adverse events also, you need to report with brand name and even the, I would say, international non-proprietary name in brackets, where you also have to put that 4-letter suffix so that you clearly differentiate that it is this product and not the other product. So that extrapolatability is not that simple. Now here is the most important slide. If you look on the left is the development pathway for an originator. It's an inverted pyramid. The biggest chunk is the phase 3 studies. Then comes the PK/PD or clinical pharmacology. Then comes the non-clinical or animal studies. And finally, the smallest chunk is in vitro studies, analytical characterization. But look at what happens on the right. For biosimilar development, the biggest chunk is right at the bottom. It's an erect pyramid. In vitro studies, CMC, chemistry, manufacturing, and control. That is the most important. If you pass that test, then only you can go on to do non-clinical studies. So which means you have to generate your own data. You can't just depend on the data of the originator. See, with the generics and small chemicals, I have seen that all that people do is take the, say, the prescribing information of the innovator, and they just change the brand name, because they don't really have to generate too much of data that they can put in their own label and say that this is my data. They just use the data of the original small chemical. But in the case of a biosimilar, you have to have your own data. Animal data, PK/PD. In the case of insulin glargine, we also had to do euglycemic, hyperinsulinemic clamp studies, bioavailability comparisons with Lantus, and finally, of course, clinical studies. And it is this totality of evidence that is looked into. And look at the clinical studies also. See, in the case of the originator, generally the studies are to show that there is a difference, drug A versus drug B, and if that difference is statistically and clinically significant, drug A gets approved. But here it is just the opposite. So with the case of the originator, what was your null hypothesis? That there is no difference. And then you do the study. If you find a difference, you reject the null hypothesis and accept the alternative hypothesis. But when you come to a biosimilar, it's just the opposite. You start by saying that there is a difference. And then when you do the study, if you actually conclude by saying that there is no difference, that means it's neither superior nor inferior. That is when you say that they are equivalent. So here, the null hypothesis is just the opposite of what you saw in the case of the originator compound. But the point I'm trying to make is in both cases, the sample size has to be adequate. It has to be adequately powered so that you can pick out the difference if that difference really exists. And if that difference doesn't exist, fine. Then you can say that this is a biosimilar. This is just, again, telling us a little more about, and this is something which a pharmacist or somebody who's a pharmaceutical or medicinal chemist would actually enjoy or know more about. I may not know as much. I mean, I know the full forms of malady, tox, etc., but this is something which is more for them. But why I'm showing you this is so that you understand that it's so complicated in terms of just physicochemical and functional characterization between a biosimilar and a biologic. Then there's in vitro functional assessments which are done, and ultimately you get this fingerprint based on form, identity, structure or sequence, size, concentration, activity, purity, and surface charge. And each of these also has some sub-bullet points. So you can imagine the kind of orthogonal analytical techniques that are used to ensure biosimilarity. It's not easy at all. Now, here I just wanted to tell you all that in the case of diabetes, all of us know last year on the 14th of November, World Diabetes Day, the International Diabetes Federation, in the 100th year of insulin, still came out with a theme, not for 1 year, for 3 years, asking the question, access. That means even now people don't have access to diabetes care. If not now, when? And why this question? At least as far as insulin is concerned, 33 million people with type 2 diabetes do not have access to insulin. And in another maybe 8 years from now, this number will go up by another 8 million. And most of them are in Africa and Asia. And this is that 3-switch design that I was talking about. I'm not sure it very clearly tells you this, but for example, recently when we got the world's first interchangeable biosimilar insulin glargine, how we had to do this study is we took patients of type 1 diabetes and they were stabilized on Lantus for about 52 weeks or 1 year. And then they were randomized into 2 arms. One arm is where they continue Lantus plus, of course, Lispro was given because they're type 1. In the other arm, interestingly, they were first switched from Lantus to Basilog for 12 weeks because HbA1c is done once in 3 months. After 12 weeks, the same patients were switched back to Lantus for another 12 weeks. And after 24 weeks, they were again switched back to Basilog for 12 weeks. So this is that 3-switch design. So now you can appreciate within the same patient, I know what is the effect of Lantus, Basilog, Lantus, Basilog. So this way, when you do your study, naturally you require a smaller sample size also because the variability is in the same patient. So I agree that there is a parallel group design also inbuilt into such a design or such a study. But in that arm A, the same patient is exposed to this kind of repeated switch. And at the end of it, obviously, you look at HbA1c reduction, you look at fasting plasma glucose because fixed fasting first is more important for basal insulin or insulin glargine. You look at the dose or the amount of insulin, is it the same as Lantus? You look at hypoglycemia anytime, nocturnal or even severe hypoglycemia. And finally, you look at immunogenicity. And in all parameters, if there is no statistically or clinically significant or meaningful difference, then only that product is said to be not just biosimilar, but also interchangeable. And this is why the US FDA on the 28th of July last year, Janet Woodcock, who normally is not so, how do I put it, voluble, she actually said that today's approval of the first interchangeable biosimilar product furthers FDA's long-standing commitment to support a competitive marketplace for biological products and ultimately empowers patients by helping to increase access. So at least to some extent, that access theme of the IDF can be mitigated with quality, world-class quality biosimilar insulin, glargine. That is just one of them, but you can have even other analogs. But ultimately, you increase access to safe, effective, and high-quality medications at potentially lower cost. So therefore, when a patent expires, now this is also important, when a patent expires, the product does not expire. But what happens, the innovator immediately wants you to switch from that product to another patent-protected product, so that at least, you know, they will lose on, say, Lantus, but they'll want you to move to, say, Tushio. But on insulin glargine, who will then do further clinical development? Obviously, the biosimilar manufacturers. They can still facilitate studies on insulin glargine. The originator company may not be too interested there. They may be more interested in that concentrated solution, which gives you more with less. And this is that naming convention that I spoke of, that 4 letter stuff, which is important from a pharmacovigilance perspective. And recently there was an article on what are the top 10 innovations. So Semglee or insulin glargine YFGN was named as one of the 10. And this is my last slide. The implications, obviously, that biosimilars, one is to treat more cautiously than generics. Physicians are obviously going to be more hesitant. There'll be some concern about purity, the risk of antibodies developing, because, clinically, how will I know that immunogenicity is developed? I can, of course, measure, but that is not done routinely, and obviously it will be a bit expensive. Long-term data we may not have. Biosimilars are not considered to be generic. So pharmacists, they expect to leave the decision of product substitution to physicians, which is good actually. The need for aggressive pharmacovigilance, biosimilars need their own international non-proprietary name. The label should be their own and not that of the originator or original reference biologic. Informed consent in some countries may need to be taken. But ultimately, the last question is not whether you can afford to prescribe either an original reference biologic or an original reference biosimilar. I know such a term does not exist, but what I meant is among the different biosimilars, are they all equally similar to the original reference biologic, or is there one original reference biosimilar which can be the benchmark for other biosimilars? Now, that is something which in future we'll have to think about. But sometimes it's not just looking at the acquisition cost, but the overall cost that can really make a difference. But certainly by using a good quality, world-class approved biosimilar can definitely improve access. So more indicated patients can benefit from that particular medicine. And then naturally they'll be more compliant, better controlled, has lesser complications. And that way the overall cost can also come down. That I think is my last slide. I'll stop sharing. I hope I've not taken too much time, but please feel free to ask me questions or just add your own thoughts, which will enrich me, as I said. Yes, Dr. Ray. Sir, you are muted.
Dr. Ray is talking, or thank you, Dr. Suvarna, for this excellent, very lucid, and you made it such a complex topic so easy. So any questions from the audience? You can directly ask Dr. Viraj.
Yeah. Sir, with all the chair's permission, may I also request today we have our President, IPS West Bengal Chapter, Professor T K Mondal, sir, and our Professor Santanu Tripathi, sir. So I request them also to have their comments too. So may I request Professor Santanu Tripathi, sir, and Professor T K Mondal, sir? Please
actually, Dr. Ray was talking, but he was muted.
Yeah, so I was muted. Now I became unmuted. Okay. So my point is, it's a fantastic talk within a very short span of time. You have covered most of the things, but there remains some debates about thinking the future of these compounds. You see, there are many terminologies: biomimics, biotopics, biocopies, then biobetters, and these. I was thinking when I was listening you, can it be like biosisters? Because these are all sisters, My point is that see, null hypothesis disproving or the null hypothesis concept is also not there when you develop it, since the topic was development of this. So that has to be. And the prescribability and switchability, these are the two important issues probably you have raised. Of course, because the cost of the medicine, you have told that it's having a lot of advanced methodology to develop this drug, though the sample size and time consumed for developing the drug was not that amount, so that we can't conclude anything. So how the cost can be minimized if the cost of production is that high? That's a question, right? And secondly, of course, it is a talk of the day. There are indications of these biosimilars is only the intractable diseases like cancers, like your autoimmune diseases and all these. But is there any indications you have found out in the treatment of infectious diseases? Like, the whole world is trembling for this COVID. So is there any clinical trial in infectious diseases about these biosimilars? So these are the few points I was thinking that it could be better to discuss. Over to Dr.
So, sir, the first question was on how would I reduce the cost of the biosimilar by reducing the cost of production, because the manufacture itself is quite expensive. So that's what I was telling you, that in the Indian biosimilar guidelines, they do not give that much of emphasis on the other aspects. That means they only tell the company at a CMC level, if you can show me that they're similar, then I will make some kind of relaxation. That means as far as your clinical study is concerned, even if it's 100 patients in each arm, it is fine. And at the most, you have to do a phase 4 on 200 patients. And again, that is fine. So that way they try to reduce the cost, because obviously if the cost comes down, then the price of that biosimilar, like if you see in the market, you'll find some biosimilars quite cheap as compared to, say, the Biocon Biologics biosimilar insulin glargine. So this is the way they at least try. And I think WHO also has the same philosophy. So I think India is following the WHO mindset. And the second question was in terms of infectious disease. So as you know, sir, even for COVID-19, currently there are these monoclonal antibodies that some companies have come out with. Now, so far, at least nobody has dared to come out with a biosimilar to that monoclonal antibody, but that could be the next step. So clearly, antibodies against infectious agents, for example, Serum Institute of India, they have a monoclonal antibody against the dengue virus. Then they have something against, I think, even the virus.
HIV, I think.
HIV also, and plus rabies also.
Yeah.
So they have monoclonal antibodies, which actually, if you look at it, some of these monoclonal antibodies, which have a very long half-life, they can do both. That means therapeutic, so they kill the virus, but then they remain in our body for a very long time. Right? There is a company called ADG, Adagio Therapeutics, and they have a particular molecule called ADG20. It's called, I think, adintrevimab or something. Now that molecule, you won't believe, it actually stays in your body for something, half-life is about 100 days. And the protection, because the level of that antibody you can measure. And you also know the level it needs to be at so that it can take care of the future. If in the future, if a virus comes into you, it can still take care of it, which means it protects you for 1 year. So initially it treats you, and then there's secondary prophylaxis for 1 year. So I feel if at all this product comes to market, it can compete with the vaccines. Because see, with vaccines, what happens is we don't know. Even with mRNA, some people are saying it is waning, so maybe 6 months or whatever. Whereas here, at least it is predictable, measurable. So as you said, I'm sure monoclonal antibodies, just like mRNA people are talking a lot about, monoclonal antibodies will definitely be one part of the future.
Thank you. Thank you. Yeah, that's the questions I had. Let other knowledgeable experts can give their opinion on that. This monoclonal antibody sometime came in during the second wave, the treatment of acute COVID, isn't it? But it didn't stand. It didn't stand.
No, sir. The monoclonal antibody, unfortunately, did not come during the second wave, and that's why we had all this problem. It came later. Actually, it's a product from Roche Regeneron, which now Cipla is marketing in India. There are others also by Eli Lilly, then AstraZeneca has come out, then Glaxo and another company, they have come out with one more. So there are about 4 or 5 which are there in the world.
But it didn't stand. WHO didn't recommend also, like remdesivir. Ultimately,
No, those are not monoclonal antibodies. Sir.
No, remdesivir is an antiviral. No, see,
Ah.
that the WHO didn't approve this thing as a treatment protocol for that. Yeah.
Dr. Santanu Tripathi, would you like to?
Yeah. So, am I audible?
Yes, sir.
Yes, I have. First of all, it's a great experience as ever listening to you. It's very well done. I have a few comments and questions. First, about you have very nicely depicted that the comparing between the generic development vis-a-vis biosimilar development. I would like your comment on, in case of generic development, there is always a allowable limit, like 80% to 125%. Okay, but when you talk of biosimilar, regulatorily, whether there is any limit, okay, or you can say some kind of allowable range, okay, even if it is 81% similar, we'll say it is biosimilar, or it is 124% similar, we'll say it is biosimilar. So something like that, whether there is any concept like that, if not, why not? This is one question. Second is about the batch-to-batch variability is not just for biosimilar, even for biologics.
Yes.
And then, is there adequate regulatory vigilance in place, or any provision for regulatory vigilance, or some kind of obligation on the part of the marketing company, okay, to provide batch to batch variability or lack of variability rather, okay, for biologics or biosimilar, and how is the situation in other countries, say European Union or US vis-a-vis in India? That is the second question. Third is next is about you have very nicely described the comparing and contrasting the development of biologicals vis-a-vis biosimilars, and you have shown that reverse triangle or reverse pyramid model. Given that it is quite acceptable globally that the amount of effort and energy to be spent in clinical development for biosimilar will be less as compared to biologics, and for it will be more effort intensive for CMC, okay, and the same model is being followed in India too, okay. So that's the other thing. That's not really a question, a comment. Your comment on the INN, international non-proprietary names given to biologics and biosimilars, particularly when you talk of biosimilars, again, if in principle, we would all accept that the biosimilars in your last, when you concluded, you have also mentioned how similar are biosimilars, okay, or how different are different biosimilars, okay. So truly speaking, in principle, biosimilars ought to be also not very similar to each other, okay. And from that angle, whether there is a case for having different INN for biosimilars or rather different biosimilars. Okay, you have mentioned that when you talk of INN, particularly if it is a prescribability, then you can have the same INN probably, and if it is the switchability is given, or rather what you have said, that interchangeability which has been established for the first product, there you say it has a right to get a different INN, but has it been really given, and whether it has been accepted by WHO, okay, for INN, this thing, international non-proprietary name. Next is about use of reference in clinical development of a biosimilar vis-a-vis biologic. But when you talk of biosimilar, it's a challenge, because why should the originator company, innovator, share the detailed references, okay, with the biosimilar development company, and how precisely that is done, and what is the practice in US or European Union vis-a-vis in India? Okay, that's one question. And even when you are doing the biologic clinical trial, what is the comparator arm for that matter? Is it just with placebo or with any other non-biologic standard treatment? The next is about collection of, when you have mentioned about that the three-switch design, so very interesting and very well understandable. I was just reminded of the N of 1 design, okay, and I find this three-switch design, okay, there must be a sample size in three-switch design study also, and there if the sample size is, say, I don't know, if it is 50, then I would consider this as a collection of 50 N of 1 study, okay, because in N of 1 study is exactly the same kind of design, okay, you keep on changing periods and then in the same patient. Coming to another question about IPR for biosimilar. Do you think there is a case for IPR or patentability of a biosimilar? Think about that. I'm making a case for that. Regarding the pharmacovigilance, yes, it's extremely important, and there has to be focused pharmacovigilance. Why our pharmacovigilance program of India is silent about that? And it is it is, I think, the need of the hour, given the fact that more and more number of biosimilars are flooding the Indian market. And finally, I liked your comment, almost concluding comment, that are all biosimilars similar to each other? And there must be good research to identify their dissimilarity or similarity. Thank you.
So, sir, now you'll have to help me because I may not remember all. So, I'll start with the first one. Now, the first one was about 80 to 125. So, as you know,
Yes.
the 80 to 125 applies to our bioequivalent studies when we compare, say, a generic to a small chemical. Now, with a biosimilar, although bioequivalence is also compared between the biosimilar and the biologic, in terms of deciding whether this biosimilar is really similar or no, what FDA or any regulator looks at is the totality of evidence. That means they look at, one is CMC, you have to prove that you're similar. Then some people also look at animal studies, they look at PK/PD, and then, as you said, a minimal clinical phase 3 kind of study where you should show that efficacy, safety, and immunogenicity, there should be no clinically significant or meaningful difference. That means statistically and clinically. So based on that, biosimilarity is approved. So that was the first question. What is the second one, sir?
Second one about batch-to-batch variability and regulatory vigilance. Need for regulatory vigilance.
Yeah, correct. So batch-to-batch variability, sir, as you know, and as I showed you also on one of the slides, abroad it is done in a very strict manner where EMA and US FDA, and I'm sure in other major markets also, the regulator from time to time will do an inspection and at random will pick up certain samples and test. So that they do, and that's how they find out if at all there is a variability. Then they reject, or as happened with the EPREX, where pure red cell aplasia actually happened and then they came to know. But otherwise, routinely they do it. Now in India, maybe the guidelines do talk about the fact that it is important to do so. And you're absolutely right that if I look at the Indian guidelines, they also give enough and adequate importance to CMC comparison. And accordingly, therefore, clinical, is not given the same. But all that I was saying is that in that clinical section also, when you're talking of that study, rather than just saying 100, maybe you make the company show some kind of sample size calculation,
Yes,
which proves,
I agree.
that's all. So it can be less than 100, can be more than 100, whatever it is. But that's all I was saying. So in India, as you know, there are rules, but whether people enforce
Implementing
these
them.
rules, the implementation is a question mark. So what is the third one?
Third one was, of course, the INN.
So, sir, INN is like this, that if you look at biosimilar, it is given a similar INN as the original biologic, but how it gets differentiated from that biologic and from other biosimilars is the 4-letter suffix. So suppose I see YFGN, that would be only with Biocon Biologics Basilon. It will not be there with Lupin or Vocard or any other company. They will have their own 4-letter suffix. So this way we clearly differentiate. And then what you mentioned about prescribability and switchability, actually what the slide said was, if you are deemed to be prescribable, then you actually need a different INN. You can't have the same INN as the original reference biologic. But if you're switchable, that means interchangeable, then you can have the same international non-proprietary name. However, still they insist on that 4-letter suffix because, as you rightly said, are biosimilars similar? That part we don't know. And currently, the regulator only says, if you're a biosimilar or if you're a proposed biosimilar, you have to compare yourself with the original reference biologic. So there is no requirement to compare these biosimilars among each other. The comparison is only with the original reference biologic. So as long as you can show that, you will get the approval. Although, as you rightly said, there could be differences among the biosimilars, but whether those differences will translate into clinical differences, we don't know. But you're right, it could be. Next question, sir.
Next question was rather a comment. I don't know whether you appreciate that regarding the N of 1 design.
Yeah. No, I agree. I had not thought of it. But now that you're saying, I do agree. And as you rightly said, in such kind of designs, you don't need too many patients. A few patients also, because it is the same patient.
Absolutely.
Yeah.
Their own control. Yeah.
Yeah.
I also made a case for IPR for biosimilars.
That, sir, is a good question, but I don't think IPR for biosimilars exists right now.
Yeah.
But maybe what I'll do is tomorrow I'll check with my people in Biocon Biologics that is there a case for at least, you know, applying for IPR. I don't know. I
Because,
don't think
why I'm saying the argument in favor is development of a biosimilar is also a hugely resource intensive affair.
Yeah.
Okay. So maybe the idea may not be very novel or innovative, but then the rest of it, okay, it is as good as creating a new molecule almost. Okay.
Correct.
So that way, I think that monopoly right.
Yeah. In fact, there are three criteria for patentability. One is novel. Second is it should involve an inventive step. And third is obviously it should have utility. That means applicable across in that. Okay.
Next comment was on focused pharmacovigilance for biosimilars.
That, sir, you have brought up a very good point, and maybe we are also thinking of partnering with PVPI. You know, whenever PVPI does pharmacovigilance workshops, if they need the help of industry, then we will offer ourselves as resources. So I know one or two people there, so I can even tell them, why can't you also focus on biosimilars, because so many are getting approved. Are doctors really reporting, and are they reporting by brand name and, you know, that INN with the 4-letter suffix? I don't think so, but let's see. I don't know.
No, it is the model also, the focused pharmacovigilance of biosimilar. The model could be also a little different from the conventional pharmacovigilance in the sense that the industry stakeholder would be very important here. And I think they can collaborate with the concerned industry. Okay. And it may not be a regulatory mandate, but then in the interest of their own product, they may be agreeing to come and join hands. Okay, that was my thoughts. Thank you very much.
Thank you, sir. Thank you so much. Any other questions, Shambo?
With the permissions of the chairman, so I, on behalf of all members of IPS West Bengal branch, heartfelt thanks to Dr. Viraj Suvarna to accept our invitation to deliver such a nice lecture at this evening. So I have no question, but two observations. Number one, what is the difference between natural products and biological products? And second, so in case of your experimental study of your biosimilar product in animal study, so we have to generate some data. So how much percentage of data generated in case of animal will be comparable with the human data in relation to your biosimilar products?
Thank you, sir. So the first question is very interesting. Now, natural products will not really involve any, how do I put it, laboratory or chemical or synthetic kind of process. Now, in a biological product, we know that it is produced by, one is our body, two is it could be a living organism. But it's not just that. In making the product, we are using some technique, some technology called recombinant DNA technology. So that perhaps differentiates a natural product, which is totally, you know, what nature really produces without any recombination. Though in nature also we can have recombination. I'm not saying no, but it is natural. It is not something where man has to actually induce that recombination. You know, like what happens in recombinant DNA technology. So that, according to me, could be the difference between a natural product and a biological product. Although biological products, as you rightly are asking, they can also be produced by nature, but then it won't be the same as what we do in the laboratory using recombinant DNA technology. Regarding the second question, sir, as I think Dr. Tripathi also said, now to make a biosimilar, I'm not going to subject that manufacturer to too much of, work, because then the price of the biosimilar will go up. I want the price to come down. So at the most, I will ask him at a chemistry, manufacturing and control perspective, if you can show that they are similar, then in some cases, they actually waive off the need to do animal data. They straight away ask, okay, PKPD, can you show me? And some people were arguing, do I even need to do a phase 3 study? So one study at least you need to do in maybe the major indication, maybe the most sensitive kind of indication as far as patients are concerned, so that, it's easier for you to show that your biosimilar works. And once you do that, then you can extrapolate from that main indication to other indications. You do not have to do studies on all the indications to get other indications in your label. This is done only so that you can reduce the price of your biosimilar so that more indicated patients can access your biologic product.
Thank you. Thank you. Over to Chairperson.
I think Professor Munshi can give some of his ideas on that because he's keeping silent. Dr. Munshi, please. He's not there. No, only one thing, just I was thinking, the most important part of this biosimilars is the part that's called purity. The purity concept, if you can make it foolproof, we can avoid the adverse effects, immunogenicity, and all these. The biosimilars, more will be pure, more will be effective, more will be safe, because the safety, chemical structure, purity, then acceptability, all these things will depend on how much pharmaceutical development to make it pure compound, and that will take care of the rest of the things. If it is true, probably future is with biosimilars, I think. So with these few words, yeah.
So may I respond?
Yes.
So I don't know whether you know Dr. Shoibal Mukherjee.
Yes, I know.
Yeah. So what he had told us, because I was in Pfizer and he was the medical director. So what he told us is that even with generics, forget about biosimilars, even with generics, although we say that you can use another process, come to the same product, and I will approve, what he said was that no matter how pure you may think your product is, it is as pure as the instrument or measurement that you're using, that lower limit of detection, as they call it. So it will detect impurities up to a point.
Yeah.
Up to a point. It cannot 100% tell you that you have no impurities. So therefore, what he said, like even in our some of these Bisleri bottles or something, when they say 100% bacteria free or whatever, it is never 100%. So what he said is that you need to therefore do studies, and according to him, even for generics, because even though you may show that they are small chemicals, they are chemically equivalent, they are bioequivalent, he's saying that no, you must do studies, and that too equivalent studies. That means generic with the original small chemical, and if they are equivalent, then yes, approve them. But naturally, regulators will not do that because they want, as I said, the price to come down. So for them, it's okay. As long as you show chemical or bio, it's fine. But he was of the opinion that it may lead to some problems. And I have experienced it because there are some doctors who have told us that when they moved from our product to a generic, their patients experience some problems. So which means even with generics, it's not 100% safe if you don't do any clinical study.
Absolutely.
No, I think, Doctor, what Professor Rai was pointing, it was the same concept. The different term has been used. By saying purity, he was possibly not mentioning about absence of any impurity, okay, absence of any contaminant. No. By saying purity, what he was trying to mean is whether it is as same as the parent product. So
no,
that
I was not meaning that. I was meaning about, see, these biosimilars are chemically large protein molecules, most of the cases. As you know, the proteins are always impure. And how much you are making these proteinaceous compounds away from this product so that we can take care of the immunogenicity, because this is the most important issue. It's not the nephrotoxicity, it's not the hepatotoxicity, it's the immunotoxicity, which is very important. So biosimilars also, see, it's very early to say because it's not time tested. It's only say 10 years old compound and concept. So what is the situation in other advanced countries about this? How they accept it? Dr. Suvarna.
They accept, how do they accept? Purity.
No. I'm talking about the concept of these biosimilars and their application and administration, not from the regulatory point of view, from the clinician's point of view.
Now they are accepting. Initially, when it came out, as you said, 2005 onwards, they were a little hesitant. And when pure red cell aplasia happened with a biologic, they were even more hesitant. But now over the years, because, naturally they know more patients can access because otherwise they're very expensive. Now clinicians, even in the US and European Union, are moving towards biosimilars, clearly. That's why these MNCs are now also into biosimilars. The same MNC which is into a biologic, they're also into biosimilars.
Okay, that's good. Anyway, long live biosimilars. That should be the theme of this today's talk. Thank you very much. Over to Shambo for the concluding part. Thank you very much from my side to you for coming and enriching us with your knowledge and all. So over to Shambo.
Thank you. First of all, we are really overwhelmed by your presentation, sir, Dr. Viraj Suvarna. Thank you very much on behalf of Indian Pharmacological Society West Bengal Chapter, and we are really enriched with the post-presentation discussion by Professor Krishnangshu sir, Professor Santanu Munshi sir, Professor Santanu sir, and finally our President, Professor T K Mondal sir. We are really thankful to all of you, and now it is almost 9:50, so we have to wrap up now. So with that, I just thanks our chairpersons for giving us their valuable time, and you, especially Dr. Viraj Suvarna sir, and hopefully we will get you in our future academic programs that will be beneficial for us. And with that, I thank you all again, and good night. And with your permission, this video, that recorded video, can we post in our that is website, Indian Pharmacological Society?
Sure. Please do. Thank you.
Thank you very much then.
Good night and stay safe.
Thank you very much, Dr. Suvarna.
Thank
Good
you.
night
Good
to
night.
all.
Good night.
Good night.
Good night. Good night.